Hello, welcome back to Honors Bio. So this is the last video in the non-Mendelian genetics section. So we walked through sex linkage, we walked through incomplete codominance, multiple alleles with the blood typing, but there's a few other non-Mendelian patterns of inheritance and things that going on that Mendel didn't fully explain. So this video is just going to go over just those last couple of lows. There's no Punnett squares, no really problems in kind of this section. It's more just different factors of how, genetics and genes are expressed in different ways. So the first way is through environmental effects. So the environment plays a huge role in how certain alleles are expressed. All right. In Arctic foxes, the coat color changes depending on the season they're in. In the summertime, the foxes have a real kind of brown coat color to them. In the wintertime, they turn white. Same thing here in Michigan with our rabbits. In the summertime, the rabbits or the bunnies have a brown fur color, and in the wintertime, they turn white. And again, that's a survival adaptation that evolved over time. Because in the wintertime, if you're brown, running around where everything's white outside, you stand out pretty easily or very easily seen by predators. All right. But in the summertime, though, if you're stark white and everything around you is brown or green, you're going to stand out pretty well too. So they evolved this so their coat color changes depending on the temperature. Same thing in the rabbits or the Siamese cats. All right, the alleles affected by temperature. So if we look here at this temperature, if this temperature is below 33 degrees Celsius, which is about 90 degrees Fahrenheit, um, the the pigment or the allele gets activated, producing this dark pigment here, which we see on the tails or the ears, or you can see it a little bit on the legs of this little kitten here. But if the temperature is above 33 degrees Celsius, then the allele is inactive, so we don't get a pigment, so it's white. So that's how the environment, just temperature, plays a role in how different alleles are expressed. Hydrangeas are another example of the environmental effects, not temperature, but really just the pH of the soil changes what the flower color of hydrangeas are, right? If the soil's acidic, so if it's a pH of 5 or below 5, we see a real deep blue color in hydrangeas. But um, as we kind of move away from the acidity and go more towards the basic side of the pH cell, we'll start to get purple, we'll get pink, and then even if it's real basic, we'll start to get red hydrangeas. So that's how like greenhouses and different like gardens are able to get different colors of hydrangeas. It's not, well, I gotta cross this flower with this flower to get this color. No, they just mess with the pH of the soil, which is going to give them a different flower color in the hydrangeas. Now, to be fair, not all flowers are like this. Hydrangeas are the only one that I know of that work with the pH of the soil determines the color of the different flowers. So that's how the environment can play a role. So the next pattern of inheritance or kind of gene interaction that we can get is called epistasis. And it's genes acting together. And all epistasis is where really one gene pair affects the expression of a second gene pair. So we see that in Labradors, in Labrador retrievers, in dogs. All right. E or gene E is gonna control gene B. Gene B is, wait, sorry, I said that backwards. E is what actually controls like the pigment of the fur, controls dog's fur color. B is what controls that expression of it. All right, so B is going to determine whether or not, E gets turned on or off, or E gets expressed. So it's similar in horse coloration because there's 2 or more gene pairings involved. So that's how we're able to get, quite a wide range of horse coloration, is because there's 1 or more genes that are controlling other genes, or they're regulatory genes. Same thing with how, between teosinte and maize, there's only 4 or 5 genes that control the difference between teosinte and maize, but they're all regulatory genes, meaning that those genes control other genes. So that's how epistasis works. So we got 2 more types of inheritance, which isn't really inheritance, it's just how are different traits just expressed. And one is polygenic inheritance. So polygenic inheritance is where multiple genes involved control a single trait, phenotypic trait. And the way to tell if a trait is polygenic is if you see a continuous variation. So meaning that if you see a wide range that normally follows a bell curve. Human height is a great example of that. There's a there's a range of differences. It's not a stark, you're either this or this, you're this or this. It's not that. People can be anywhere from below 5 feet all the way to, 6'3", 6'4", 5. So it's this polygenic inheritance. So if we see this continuous variation or a gradient appears of different options, then it's a polygenic trait. Hair color and eye color are examples of polygenic inheritance as well. Multiple genes affect hair color and eye color. So what we see is a wide range of hair color and eye color, right? We can have hair color all the way from a real dark jet black all the way to like blonde, sunshine blonde, like real light blonde hair, and everything in between. So that's polygenics. So polygenic inheritance is where multiple genes affect one trait. We can have the opposite occur too, where one gene affects more than one trait. And we call that pleiotropy. And that's all it is. A lot of diseases work that way, where multiple symptoms can be traced back to one defective allele. Cystic fibrosis and sickle cell anemia are great examples of that. So in cystic fibrosis, right, we get lung congestion and malabsorption of nutrients by the pancreas. So that's at least two different phenotypic traits that are traced back to one allele, right? Sickle cell anemia is the same way. Huntington's disease is the same way. One allele has more than one effect on phenotype. So that wraps up the non-Mendelian inheritance part of this. So we worked through sex linkage, we worked through incomplete dominance, codominance, multiple alleles with the blood typing, and then the last little bit of this was just kind of like the different forms of inheritance with the pleiotrophic, polygenics, epistasis, and then the environmental effects. So that's it. If you have questions, let me know, and I'll see you next time. Bye.